Hi everybody and welcome to the second lesson in section 2.7, which is looking at the genetic control of metabolism. And in this lesson we're going to be looking at recombinant DNA technology. So there were two processes that we could use to improve our strains of microorganisms. We looked at the other one last lesson, which was mutagenesis, and this lesson we're going to be looking at recombinant DNA technology, or as you more commonly know it, genetic engineering. So by the end of the lesson, I want you to understand the reasons why we are carrying out genetic engineering of microbes, describe the steps or recap the steps in the process, and then also identify the features and limitations of plasmids as vectors within the process and have a look at what the alternatives are. So, why are we using recombinant DNA technology or genetic engineering? The whole point of it is it can be used to give microorganisms genes to improve them, and those genes could be genes that amplify metabolic steps or remove inhibitory controls and therefore increase the yield of the protein that we're trying to get from our microbe. It could be a gene that causes our cells to secrete their products into the surrounding medium, and that means that we can extract it easier when we're growing them in a fermenter. Or it could be a gene that prevents the organism surviving in an external environment, and we put that in the microbe for safety. So it might be dangerous for this microbe to be existing in an environment, so we put in a gene that stops that from happening. So artificial transformation using recombinant DNA technology is more commonly known as genetic engineering. Now what we're going to do is we're going to recap over the steps for genetic engineering, but you might want to pause the video right now and write down what you can remember about genetic engineering from National 5 Biology. So what are the steps that are taken in order to produce genetically modified microorganism? And remember, the example that we looked at in National 5 is we looked at engineering a bacterium to produce human insulin. So can you think about the different steps? Can you think about what enzymes were involved? Have a little pause of the video and, have a little blurt of what information you can remember. So, artificial transformation is the transfer of genes from one species of organism to another, and that enables our second organism to produce a specific protein. So examples that we can have are things like enzymes or hormones, and the example that we looked at in National 5 is obviously we looked at our bacteria producing the hormone insulin that can be used to treat diabetes. Now, there are very simple steps in genetic engineering. Firstly, the gene for a desirable characteristic has to be identified. So within the whole human genome, remember 20 to 25,000 genes, we have to find the gene that we need that's going to create the protein that we're trying to produce. So we have to select that first. We then have to cut that gene out of the whole genome and then splice it into the DNA of a vector. A vector is simply something that's going to transport our gene of interest into a new host cell. And in this case, we're looking at engineering microbes, so it's going to go into a bacterial cell. So what we'll do is we'll recap over each of these steps in a bit more detail. So plasmids are vectors, and what that means is they are used to carry the information from the genome of one organism into another. So at National 5 level, we looked at plasmids that were used to insert our gene of interest into, and then we put the whole plasmid that contained the gene into a new organism. However, they're not the best to use, our plasmids, and actually what we tend to use is artificial chromosomes, because we can carry a lot larger fragments of foreign DNA in an artificial chromosome than a tiny plasmid. So artificial chromosomes are used preferably to plasmids because they can carry more genetic information. You'll see why we need to carry more genetic information as we go further through this lesson. And that's just showing you a picture of the plasmid. So remember, we've got our red structure there, which is our gene of interest, and we splice that into the green structure, which is our plasmid that we've cut open, to form our recombinant plasmid. And just to recap, we use enzymes in this process. So restriction endonuclease is the enzyme that is used to cut, so it's the chemical scissors that are used to cut open both or cut out the gene from the genome, and also to cut open the bacterial plasmid at something that's called a restriction site. But we're going to add a little further detail to this. So this is our genome, and you can see that blue section is the gene of interest. So that's the gene that's going to produce the protein that we're trying to get our microorganism to produce on a large scale in a fermenter. So we use a restriction endonuclease enzyme to cut out that required gene, and what that enzyme will do is it cuts at specific base sequences. So it's not actually scissors, it's an enzyme, and it's going to cut at either side of the gene at a specific base sequence. Now what that does is it produces a sticky end. Now what I mean by that is we've cut the DNA at a specific base sequence, so it might be ATT. So what you can see is this pink side here is a single strand of bases, and it might be ATT. Now the reason why it's called a sticky end is because, as you know from Unit 1, our bases will form complementary base pairs. They'll form hydrogen bonds between complementary base pairs. So that ATT would quite happily form a hydrogen bond with the sequence TAA. At the other side, because we've used the same restriction enzyme here, we have that sequence TAA, and again that's called a sticky end because it will quite happily form a hydrogen bond with the complementary base sequence ATT. So that's what a sticky end is. Now we use exactly the same restriction enzyme to cut open our plasmid. So again, this restriction enzyme is going to cut where it sees the sequence ATT. So we cut open that plasmid, and again we have a plasmid with some sticky ends. So at either end of the plasmid here, we'll have an open single strand sequence that it will quite happily form hydrogen bonds with the complementary base sequence. So at either end there'll be the sequence ATT or TAA, and so those sticky ends that are here will quite happily form a hydrogen bond with these sticky ends here. Okay, so there's lots of different restriction enzymes. They all cut at different base sequences. It's important to use the correct one when you're locating your gene of interest. You obviously need to use a restriction endonuclease that's going to cut a base sequence that you find at either end of the gene, and then remember to use the same restriction endonuclease to cut open your plasmid, so that your plasmid and your gene of interest have complementary sticky ends. And this is what it looks like in a bit more detail. So you can see here we've got our double-stranded DNA. The restriction enzyme that we've used has cut at the base sequence AATT, and so it's left open those sticky ends. So that AATT sequence will form complementary hydrogen bonds with the base sequence TTAA. Once you have cut out your gene of interest and you have cut open your plasmid with your restriction endonuclease enzyme, then yes, that bit of gene will actually, find itself joining in with the plasmid just because of the complementary sticky end. So the hydrogen bonds will form between those complementary base pairs. However, we need to seal the DNA. We've also cut between a phosphate and a base, so we need to use the ligase enzyme that acts like your chemical glue to seal that sugar phosphate backbone and make sure that that gene of interest is sealed within our plasmid, which remember is the vector that's going to carry our gene into our new organism. So then what you do is then you would get that plasmid and then you would insert it into your bacterial cell. And you'd insert it into a bacterial cell lacking plasmids. And then that bacterial cell, as it divides, will hopefully replicate your plasmid, and then it would also replicate that gene of interest. And that gene of interest would be switched on to produce the protein that you need, and then secrete that into the growth medium for you to extract, purify, and then sell. So transformation is all about putting your recombinant plasmid, so that's the plasmid that contains the foreign gene, into a new cell. And in this case, because we're looking at microorganisms, it's going to be a bacterial cell. So this is just showing you the whole process from beginning to end. So you can see at the top there, step 1, we've got our human cell and the DNA has been extracted from the cell, and then restriction enzymes have been used to cut out our gene of interest from the whole genome. At step 2, you can see that the plasmid, which is our vector, has been removed from a bacterial cell. We then insert our gene of interest into our plasmid and seal it with the ligase enzyme. That recombinant plasmid is then placed into a bacterial cell that is lacking a plasmid, and then that bacterial cell will multiply and grow, and as it multiplies and grows, that gene will be expressed, and it will produce the protein, which can then be extracted, purified on a large scale. So what we're going to look at next is we're going to look at effective vectors. Now, what I've got a picture of here is something called the pea-glow plasmid. As you remember, the plasmid is our vector that's going to carry our gene of interest into the new organism. Now, it's not just the gene of interest that we need to have in our vector. So you can see within this pea-glow plasmid, we have something called the GFP gene on the bottom right-hand corner. This stands for the green fluorescent protein. Now, if we were doing this back at school, we would get you to do a practical whereby we would get you to insert this particular plasmid that contains the green fluorescent protein into a bacterial cell, an E. coli cell. The green fluorescent protein has come from a jellyfish, and it's a gene that produces a protein that fluoresces under UV light. And so what we're going to do is we would take this plasmid, use the technology available and all the processes to insert this plasmid into a bacterial cell, and then check to see whether it's produced that protein by shining a UV light on it. However, you can see there's lots of other features on this plasmid. So there's something called RSC, there's something called ORI, and there's something that's called BLA. And we're going to talk about what the different features or what different features need to be on an effective vector if we're going to carry out this genetic engineering process properly. And this is what it would look like in your lab. So you can see these E. coli cells here have been transformed with the pea-glow plasmid. So they've taken up that pea-glow plasmid, and then as they've divided and grown and formed this big colony, they have expressed that protein, and that's why these E. coli colonies here are glowing under UV light. So the first thing that we need on our effective vector is we need a restriction site. And a restriction site is just the place where the restriction endonuclease is going to cut. So we've got that opening where we're going to add our target sequence of DNA. So where that GFP gene is, there would have previously been a restriction site, so a sequence of bases that could have been cut open that formed those complementary sticky ends to the base sequence either side of that gene that is being inserted. So in this case, the green fluorescent protein gene. Now, the other feature that we're going to need in our plasmid is we're going to need an origin of replication, and that's indicated on that picture by the ORI. Now, these particular genes here are genes that control self-replication of the plasmid DNA. So it's really important that when our bacterial cell replicates, that our plasmid's going to replicate. Also, if we have lots of different copies of the gene, then that means we're going to get more product from fewer cells. The other thing that we're going to need in the plasmid is we're going to need regulatory sequences that are going to control gene expression. Now, in more simple terms, what that means is we're going to need genes that are going to turn on our gene of interest. And here on the pea-glow plasmid, the regulatory sequence is that RSC. Now, this particular gene sequence here ensures that that GFP gene is only switched on in the presence of arabinose sugar. So it's only if those E. coli are grown in the presence of arabinose sugar that that GFP gene is going to be switched on. It's going to express the GFP protein and then fluoresce under UV light. And just to show you that from the results here, so we've got 4 plates here that we have, inoculated with E. coli that has taken up that pea-glow plasmid. Ignore the first 2 plates where it says minus DNA, because those 2 plates are controls and they're growing the E. coli bacteria that hasn't had the plasmid inserted into it. Now the bottom 2 plates here, you can see on the left hand side we've got a agar plate that contains the E. coli, that contains the plasmid, that contains the GFP gene. But on this plate here, we have no ARA, we have no arabinose sugar. So even though we can see there's loads of bacterial colonies there growing on that plate, they are not producing the GFP protein. And we know that because it's not fluorescing under UV light. And the reason they're not producing that GFP protein is because there isn't the arabinose sugar there. And remember, we need the arabinose sugar to switch on that GFP gene to enable it to produce the protein. And that's what you can see on the right hand side. So you can see where it's plus DNA, so bottom right, and we've got a plate that says LB AMP ARA. LB is just the name of the agar that we're growing the E. coli on. The ARA indicates that that agar has arabinose sugar within it. Now, because we've got the arabinose sugar within it, that is being taken up by the E. coli cells, and that external factor is switching on that GFP gene to produce that that green fluorescent protein. So that's why these particular E. coli here are glowing under UV light. So it's just to give you an idea about what a regulatory sequence is. It's basically a switch to turn your gene on or off in certain circumstances. Remember, with genes, they can be present in the cells, but we need to have either the internal or external factors to either switch them on or off. Now, another feature that we need to have within our plasmid is we need to have something called a marker gene. Now, a marker gene is needed to see which cells have been successfully transformed. Now, I want you to look at this diagram at the top of this page. So you can see we've got our restriction enzyme that's used to cut open our plasmids at the very top, and then we've got our restriction enzyme that's used to cut out our gene of interest. And then remember, what we do is we insert that gene of interest into a plasmid. And the reason that they join together is because of the complementary sticky ends. However, there's 3 outcomes that can happen. Remember, we've got loads of like gene of interest in a sort of test tube, let's say, and then we've got loads of our plasmid in another test tube, and we're just simply going to mix them together and hope that the gene of interest sticks into our plasmid, and then we seal it with the ligase. However, 3 outcomes could happen. Either because they have complementary sticky ends, all the gene of interest could like join together and form just like a loop of like the gene of interest, which you can see at the very bottom that says circularized DNA. Or what could actually happen is the original plasmid could just seal back up without any gene of interest in it at all. So we've just gone back to a plasmid that does not have our gene of interest in it. Or what we want to happen is our gene of interest could insert into the plasmid and we have a recombinant plasmid, and that's what we do want to have happening. However, we don't know, okay, so that's what we've got in the test tube. We've got these 3 outcomes, and we're then going to transform our E. coli cells. And so our E. coli cells are going to be, put under a technique to make them take up these plasmids. Now, we don't know which cells have got the plasmid that has got the gene of interest in it. We don't know which cells have taken up just circularized DNA. We don't know which cells have just taken up the original plasmid. We do, however, when we use what are called marker genes. Now, these are usually antibiotic resistance genes, and the antibiotic resistance genes allow us to see which cells have taken up the recombinant vector, so that we're only growing that. It would be a total waste of time, money and energy if you were growing all of those cells in the fermenter, and only 33% of them are actually the ones that are going to produce your protein that you're after. So marker genes allow us to see which particular E. coli cells have taken up our recombinant plasmid. So I've just given you an example in the picture above here. So in this particular example, the plasmid has a marker gene that gives the cell resistance to tetracycline. So tetracycline is an antibiotic that would normally kill off our bacterial cells. That particular plasmid that also contains our gene of interest contains a tetracycline resistance gene. So you can see we've got our plasmid vectors in that first picture, and we've got our bacterial cells without the plasmids. We've carried out a technique to make sure that our bacterial cells are taking up the plasmid. However, some cells don't take up the plasmid at all, and only a few cells will actually take up the plasmid. What we then do is we grow them on an agar plate containing tetracycline. Now, those 2 cells at the top that don't have the plasmid, they don't have the tetracycline resistance gene, so they're not going to grow. They're going to be killed. So we know which cells to grow in our fermenter because they're the ones that will grow on this particular plate. We don't have to use selective markers like this, which usually are antibiotic resistance genes. The other thing we can use is we can use a screenable marker. So actually a GFP gene can also be inserted as a marker gene, and then we can simply shine a UV light on our bacterial cells, and obviously the ones that glow are the ones that have our gene of interest and can be used to then make more. And again, going back to our example from the P-glow activity that we normally carry out in class, we have got 4 plates here, and again, but on this particular plasmid, the marker gene is a gene that gives the bacterial cells resistance to an antibiotic called ampicillin. So at the very top 2 plates here, you can see that those 2 plates there contain or we've inoculated those 2 plates with E. coli that do not have the plasmid at all. Now, on the left-hand side, that particular agar plate just contains the nutrient agar. The E. coli will grow quite happily because there's nothing killing them off. On the right-hand side at the top, our E. coli cells haven't grown at all, and that's because those E. coli cells that do not have the plasmid do not have the ampicillin resistance gene, so they've all been killed off. Okay, whereas at the bottom, on both plates, we've got the E. coli that contain the plasmid, and therefore those E. coli have the ampicillin resistance gene. So we've grown them on agar plates that contain the ampicillin. So you can see on both of those plates at the bottom, it's labelled AMP, AMP. So therefore on both plates, the cells will grow. So that's an example of a marker gene. And the last thing you might want to have in your vector is you often want a gene that will prevent the survival of the microorganism in an external environment. Okay, we don't often want our modified bacteria going to the external environment and passing on those genes through horizontal or vertical transmission. So we put in a gene to prevent their survival outside of the lab. Now you'll see now why a plasmid maybe isn't quite suitable for genetic engineering. Our vectors that we're going to use to transform our bacterial cells need to contain a huge amount of information. They need genes for the origin of replication, they need regulatory sequences, they need marker genes, we need health and safety, or genes that will prevent our microbe surviving out in the environment for health and safety. We also need to put in our gene of interest, and this is why rather than actually using a plasmid, we use an artificial chromosome that will contain all of those things and be able to carry much larger sequences of DNA into the bacteria. So the next thing we're going to look at is what we actually transform. Now, unfortunately, using prokaryotic cells like bacteria also has limitations. Our prokaryotic cells don't carry out what are called post or pre-translational modification. When a gene is expressed, remember the gene undergoes transcription and translation. So that gene is first transcribed into messenger RNA, and then that messenger RNA will then translate into a polypeptide chain. A polypeptide chain is not a functional protein. Remember, what has to happen is that polypeptide chain has to be either cut, or it has to have things added to it, or it has to be then folded to make a fully functional protein. Now, the prokaryotic cells are often unable to make active proteins because they can't carry out those modifications, and often it results in our polypeptides being incorrectly folded. So rather than actually using a prokaryotic cell, again for genetic transformation, we often use a yeast because that is a eukaryotic cell and it can carry out those post and pre-translational modifications to make sure that we're getting a fully functional protein and not a polypeptide chain that's completely useless. Right, so we've covered a large amount of information in that lesson, but what we're looking at is how to improve strains of microorganisms, and this is a technique used to do that, our recombinant DNA technology. Now remember, what we're looking for you to take from this lesson is to identify the reasons for carrying out our recombinant DNA technology. So what are the 3 reasons why we might improve a microorganism? To describe the steps in recombinant DNA technology, and that is very much the same as National 5, with the only extra bit of information being about the sticky ends produced by our restriction endonuclease enzymes. And then lastly, identifying the features of the plasmid. So what are those extra genes that we need within the plasmid to help our gene of interest be carried into our target organism, and then also the limitations of plasmids and the alternatives that we can use instead of that. So I've put a couple of little learning activities in the Google Classroom for you to have a go at, and good luck with that. And remember, if you've got any issues, then please don't hesitate to email us.